Yeah, and we'll let you know. Yeah, I don't know, I still have to take
Okay, guys, so nobody is here today. So what we'll do is I'm going to give you guys all the answers to the test. Because you came. That's really nice of you. How many of you guys are taking the exam today? Thank you. I appreciate that. A, B. I couldn't do that if I wanted to, right? Because it randomizes. Okay. So quickly, obviously this week's the exam number 2. A quick note, so I've had a huge number of people requesting to take the exam on Thursday instead of Tuesday. And I'm pretty flexible. I'm like, I don't care when you take the exam, whatever, just take it. The problem is, now so many people have opted to take it on Thursday, that it's going to be like a freight car. It's going to be like a cattle car. You guys are going to be like sitting like this. And it's going to be like type in your ear. I'm sitting on the floor with someone sitting on my lap. It's going to be a disaster. So to avoid this from happening next time, I'm going to offer a bonus to people who take it on Tuesday. Because I know there's this feeling of it's not fair, I want more time, and I don't want people to feel that things aren't fair, so I'm going to offer a bonus. Yes, if you normally take it on Tuesday, you get it. It doesn't mean you get 5 points for showing up on Tuesday. It means there'll be an extra credit. Yes?
Do we have a bonus today?
No, because that's not fair to the people who already got permission, right? Like, what if you were like, oh my God, I'm going to take it on Tuesday, and then you found out that your whole group got a 10-point bonus. You'd be like, that is not fair. Like, the world is not fair. There's no way to make everyone happy. But I think I'm going to try some kind of motivator so that the people who feel like it's not fair they have to take it Tuesday will feel that it's more fair because they get an extra opportunity for more points. That's how we're going to work the next exam, because it's really going to be an ugly situation on Thursday. Those of you guys who are taking it today, you're going to get to spread out, put your feet up. I'll be passing around martinis. Good. Okay, so let's take our 5 minutes to write. You might say to me, whoa, we haven't learned anything yet. But I want you guys to just take a minute to kind of take stock of what you already know. Yes, go ahead and get your notebook out. I know everyone's so excited about the thought of taking the exam on Tuesday next time. That's a different story. So, how are we doing? You doing good? You doing good? Everyone doing good? Okay. So let's talk for a minute. I know it's hard to focus when the exam is coming up, but it's good to clear your mind and not think about signaling. So much chatter this morning. Not think about signaling for a minute and think about the cytoskeleton. Clear your mind. It's like yoga for your brain. Okay, so I want you to just take 5 minutes and talk about the cytoskeleton. What do you know about it? What are your current understandings of the cytoskeleton? What are the major components of the cytoskeleton? When I say cytoskeleton, what proteins do you think of? What cellular processes or functions are dependent on the cytospace skeleton? What is that? I'm telling you, you don't even want to know what the exam looks like since I was up till 1 writing it. Okay, so take your 5 minutes and then we'll discuss and then we'll actually learn something. Okay, guys, I'm cutting the 5 minutes short because there's so much chitter chatter, and I know people are distracted and it's hard to focus, and so it's not really serving its purpose this morning. But let's talk, okay? So I saw some people had Wikipedia open on their computers, and you know, that's fine because you're kind of refreshing your memory and learning a little, and it's not cheating, it's actually teaching yourself something, so that's always fine. Okay, so I'm gonna call names because it's gonna be fun to see who's here. Savannah, I saw Savannah. What do you know about the cytoskeleton?
Well, because I have the test on the brain, I was relating it to the test today with the Rho protein.
Right.
And how in the presence of a ligand, it causes the actin to stop growing, so the growth curve stops. And without the presence of a ligand, the Rho is growing, so the actin is growing and causing the cell to move, which changes the cytoskeleton.
Okay, so that was very smart. Yeah, so Rho, and we will learn, okay, so I heard keywords of actin and Rho, and so we're gonna learn all about this. You guys have had that preview for about a week now about actin and how the conformation of actin is related to cell movement. And that signaling actually controls us also, in addition to controlling gene expression. Very good. What else do people know about the cytoskeleton? Yeah. It's important for the structure of a cell. Yep. What else? Yep. Microtubules are involved with movement of things within the cell. Yep. Okay, so cytoskeleton is like a scaffold, keeps things together, allows things to move around within the cell, keep things organized. What else? Okay, so motor proteins, they move in different directions along the cytoskeletal components, specifically actin and microtubules. What else? Let's see. Yep.
Cell division depends on the expansion of the cytoskeleton.
Cell division is dependent upon the expansion. I'm going to change that to the organization of the cytoskeleton. Absolutely. What components, what major structure involved with cell division is made up of cytoskeletal proteins? Mitotic spindle, that's great. Everyone says mitotic spindle, fantastic. Yay, 201 instructors. Good. What other cellular processes? So we said something about some processes. What about what are the major components of the cytoskeleton? Yep. Actin. What else? Yep. Myosin, right? And so that's one of these motor proteins. Yep. What else? Yep. Tubulin, which make microtubules. Yep. What else? There's one that I pay almost no attention to, but it exists, and it's important for strength of cells and formation of organs from cells.
Centrioles.
Hmm?
Centrioles.
Intermediate filaments. Yep. And so there are some other motor proteins that we'll be learning about. So one with her is myosin, kinesin, dynein, all motor proteins. Remember in the beginning of the semester I showed you guys a movie, and then in the movie there was a motor protein holding onto a vesicle, walking along microtubules. And so that's pretty much what we'll be learning about, the formation of these structures and the motor proteins that walk along them and things that they move, be it a chromosome or a vesicle or an organelle. What else does the cytoskeleton play a role in? We talked a little about it with Roe. Movement, right? Some cells crawl, some cells move. And so that's controlled by the cytoskeleton. So I think before I had to start teaching this, I never really did too much with cytoskeleton. I didn't know too much about it, and other than what you guys know, pretty much. And I was like, oh, I better read up on it since I have to teach it. And I really thought it had to do with just keeping the cell shape. I didn't realize how important it was. So we're also going to learn about all these different accessory proteins that help to regulate the dynamics of the cytoskeleton. Okay, very good. Thank you for your participation. Okay. So this is what we were just talking about, right? What is the cytoskeleton? So this right here is immunofluorescence, and we've got three things stained, right? You see blue, you see green, you see red. Blue is DAPI, it always stains DNA, so those are your nuclei. Red is staining actin. Notice it's around the edges of the cell, around the cortex of the cell, and it's that change in actin around those edges that's involved with movement. Green, you notice it's everywhere, right? So knowing what you know about microtubules and the fact that there's no organization really for these microtubules, there's some organization, but would you say that this cell is a mitotic cell or an interphase cell? Raise your hand if you think it's a mitotic cell. Each one, they're all the same, obviously. Raise your hand if you think it's an interphase cell. Right, interphase means not mitosis. Right. So, right, exactly. So how would that look if it was a mitotic cell? So you have it, what would the green staining look like? Different maybe? Huh? You'd see the mitotic spindle, because the mitotic spindle is made of microtubules, right? So it looked like little starbursts on either side, right? Also, your DNA would look different, right? It would be condensed, it would look like chromosomes. They'd probably be lined up in the center. So I think I have some pictures of that coming up. So we'll just have a little bit of inspiration today. Right? You need inspiration to learn something new, right? So I'm going to inspire you to love the cytoskeleton. I really didn't care about it, and then as I started to learn about it in great detail, I was like, this is so cool. So then again, like that's me being a scientist. So here's mitosis, right? So we're talking about interphase. Notice you've got your so again, the same staining, right? Blue is DNA. Red, in this case, is mitochondria. You'll notice they kind of move around a little. And then green is microtubules, which is really what we're tracking, our DNA and our microtubules here, because this is mitosis. And so notice in interphase, you don't have that condensed chromatin, and then you've got just interphase microtubules. But in prophase, you start to form your mitotic spindle. And so you see complete reorganization of your microtubules. So you have to think to yourself, wow, they're reorganizing, that's crazy. Microtubules must be really dynamic. They must change really easily. And that's what we'll be learning about. And so you see your mitotic spindle is going to bind to your chromosomes and line them up in metaphase. And then in anaphase, you get the chromosome segregation. And so notice your microtubules kind of attached to your chromosomes, pulling them apart. And then interesting here is in telophase and cytokinesis, you can't see it here because your red is your mitochondria. But if we had stained actin, we would see that a contractile ring forms in telophase, and it contracts, and that's what kind of pinches the cells into two. So I would say that the cytoskeleton is important for mitosis. I would say that. So then there are different, there's kind of like a language you have to learn to study the cytoskeleton. And so there are a number of phrases that will kind of come up, and I just want you to learn and get used to them so you know what we're talking about. So one is dynamic instability, right? So what does dynamic instability mean to you? Just the words. Dynamic instability. Changing and not stable. Exactly. And that's the exact term to think about with the cytoskeleton. Your actin and your microtubules, they're not stable and they're changing constantly. They're these subunits and they assemble, so we call that polymerize. They come together into a polymer, so actin filament will grow and then it will disassemble and it will shrink. The subunits will move and go to where they're needed, and so the term to think of is dynamic instability. And so what this is showing, this is a time-lapse image. So each picture is taken at a different time, increasing time, and it's following GFP-labeled microtubules. So each line is a microtubule, and what you'll notice is they're changing in length. Right? You know, here's shorter, here's longer, here's shorter, here's longer, here's shorter. And so what's happening is the microtubules are constantly growing and shrinking. And that's dynamic instability. I feel like I have a movie for you that's going to show it. So this is showing microtubules. And so here's your centrosome, and that's where your microtubules kind of, you can think of it as where they grow from, and we're going to learn about that in detail. And let's see what I can do here. So that's going to show you, here's your centrosome, and those are all microtubules kind of radiating out. And so notice they're growing and shrinking and growing and shrinking. That's dynamic instability, constantly growing and shrinking. And this is important to, as you'll see, this is important for understanding how the cytoskeleton works. So we're talking about what these different cytoskeletal components are important for. Well, like in that movie we saw in the beginning of the semester, they're very important for movement of organelles and vesicles, all that stuff we just learned with vesicular transport, that's happening by motor proteins binding to vesicles and walking along either microtubules or actin, depending on the situation. So I believe I have another thing for you to see here. I'm always saying a prayer that what I want to happen will happen. Okay, so this, let me make it big. So what you're seeing there, oh, and that was that. Let's try again. So you're seeing mitochondria moving around along microtubules. That's so fast. And it's showing how the virus is inhibiting that. But if we go back, we're going to keep watching it again and again so you can really catch it. But what you're going to see is the mitochondria are going to be bound to motor proteins and walk along microtubules in the neurons. You can see them going up and down. Right? Everyone see that? And then with the infected neurons, there's a protein in the virus that's preventing that from happening. Don't worry about the infected part. I just want you to understand the concept of the microtubules are kind of acting like tracks in the cell that things will move up and down on. I love movies. They're fun, right? Yeah, a little bit. Okay, so you guys have access to all this and more. I feel like my computer and the system are having a all the time. What's up there? It's gonna happen, guys. See, aren't you glad this isn't like this super fast-paced class? Okay. It's coming, I promise. There you go. Huh? I am recording it. Yep, I'm at 22 minutes. We're gonna be okay. Okay, so organelles. So just what I showed you is organelles moving along these tracks using organelle-bound motor protein. And so there's another movie here. We're just gonna mess with my computer all day here. And so this should show, this is embedded, so it should be a little bit better. And so here what you can see here is a microtubule, and you're gonna see that organelle kind of moving along the track. You see that? Oh, there goes another one. Oh, there goes another one. And so you said plus, you see it plus n and minus n. And so that's because there's polarity to microtubules and to actin, and different motor proteins walk in different directions. And we're going to learn about that. So if a motor protein needs to walk in one direction, if something goes in one direction, then it's usually bound by a specific motor protein. And so we will learn all about that. Why do organelles need to move? Because they need to be able to do. Is that a good one? Well, so the best example that I have is always more thinking about vesicles, because we know why they need to move. It's very clear that something needs to go from one place to another. And so organelles are going to move during the cell cycle. Have to come up with a better answer for you. Okay, so now we've got vesicular transport. So you can think of the vesicles traveling along cellular highways. And so here's a microtubule with two different motor proteins that go in two different directions, dynein and kinesin. And this is another movie. This goes. My computer is going to just jump out of a window with all this. Oh, awesome. Oh, God. Let's make this stop. Go away. You know what would be really cool is if I was being observed right now. I'm very nervous that I'm going to screw something up here. Okay, let's see. Dr. Ehrlich went crazy in class. It was so weird. It was kind of scary. Okay. Let's see what. I bet. Do you guys think it's going to be moving or it's going to be stationary? Let's take a vote. Okay. There you go. You guys will have to look at that video on your own. So we've been talking about microtubules and motor proteins and vesicles moving and organelles moving. So here, so let's talk about actin and cell movement. Right, so this is, I'm gonna mess with it again. This is a neutrophil. A neutrophil is a white blood cell. And it's gonna be, so the neutrophil is right here. And all these dots are bacteria. So these are all, all these dots are red blood cells. And what we're gonna see, can I see it here? Right there, see those little dots right there? There, the resolution's not so great. That's Staph aureus. So let's see what the neutrophil does. So there's your neutrophil. Those are red blood cells. Oh, that's what I just told you. Okay, there's your bacteria. And so the neutrophil wants to get the bacteria, right? Yeah, and so what it's going to do is it's going to chase it until it catches it. And then what it ends up doing is phagocytizes it, which we should see shortly. Oh, God. It's going to get it. Oh, got it. And I believe there's sound effects with the movie, and there's a burp at the end. I'm pretty sure. We'll have to see. That red blood cell doesn't look so good. So all that movement is controlled by actin, and that in disassembly of actin in one direction of the cell and assembly in another of the cell, which is controlled by a Rho. And constant growth of a web of actin is going to push that cell forward. Okay. Whoa. Okay. So let's talk a little bit more about these components, right, of the cytoskeleton. You've got green microtubules. Oh, I didn't say anything. Nothing important. Green microtubules, blue DNA, and then you see red actin around the edges. So that's the cytoskeleton that we're going to be focusing on. So what role does it play in complex cellular processes? Cells need to interact with the environment. That's the mantra of the class, right? With signaling, you're mounting a response to the environment in some way, right? Well, one of those responses, as we saw when we were talking about axon growth and growth cone collapse, one of those responses is the shape of the cell, the movement of the cell. It's not just gene expression, right? We just saw a neutrophil that was running all over the place to chase bacteria. That's a controlled, that's not random, right? That's regulated. That's a regulated response to the environment. They need to change shape and move from place to place. Cells need to grow. They need to divide. They need to respond to extracellular signals. Pulling chromosomes apart at mitosis, that's very dependent upon cytoskeleton. Driving intracellular traffic, we just saw that with the vesicular transport and movement of organelles. Without the cytoskeleton, sperm couldn't swim. That could be a problem. And white blood cells, the neutrophils that we just saw, they need to crawl across surfaces so they can get to whatever they're trying to get. And then, of course, for those of you who took A&P, muscles need to contract, and that also requires cytoskeleton. Question? No. So, did I convince you it's all very important? Yeah. Okay, so here are the key players of the cytoskeleton. You guys already know this. Intermediate filaments, so they provide mechanical strength, resistance to shear stress. Microtubules, involved with positioning of organelles. They direct intracellular traffic, form the mitotic spindle, act in filaments, cell surface shape, movement, or motion. And then there are all the proteins that control them. Here are these accessory proteins that link cytoskeletal filaments to organelles or control the function of the above proteins. And so that's where things are going to get very confusing for you if you let it happen. And I have to tell you right now, as we go through, so basically what's going to happen is we're going to talk about actin, we're going to talk about microtubules, we're going to talk about, and then we're going to talk about a whole bunch of proteins that regulate them. And so that's where you're going to be like, oh my god, all this detail. And what I want you to do right off the bat is draw yourself a picture of actin and tubulin, and then add in each component, each protein that regulates it, along with kind of like a little definition of what it's doing. One protein might sever microtubules, one protein might stabilize microtubules, and then you'll have your microtubule, and then you'll be like, sever, So it'll help you to kind of keep yourself organized. Also, there's a really nice 2-page spread in the book, which does exactly that. It shows your microtubules, it shows your actin, it shows all the accessory proteins and what they do. So just keep that in mind. So the cytoskeleton is dynamic, right? There are a lot of different changes constantly happening with the cytoskeleton, such as cell division, right? So normally when you're interphase, you're going to have, you might be a crawling fibroblast, and you'll have a polarized dynamic actin cytoskeleton. What does that mean? Polarized, it means it has direction to it. It's dynamic, so that actin is growing and shrinking, or growing, and you're having actin leaving from one end and adding to the other end, pushing the cell forward. And then your microtubules, they're interphased, so we're not actually in cell division at this point, so they're just organized at the centrosome. But during cell division, the microtubules are going to reorganize to form your mitotic spindle, preparing to segregate the chromosomes. And then you're going to get the actin reorganizing. It's moving from being involved in cell movement or reorganizing to form a contractile ring. And so it's going to squeeze tight and separate the cells. And then you have 2 cells, and it's going to return to that original state. So dynamic, constantly changing, a lot of changes associated with cell division. Questions? So these cytoskeletal filaments are constantly changing in response to stimuli. Reorganizing during cell cycle, and this reorganization is occurring through growth and disassembly of subunits. Right, so here's your microtubules at interphase. You've got your centrosome right near the nucleus, and that's where all of your microtubules are going to radiate out from. And then during mitosis, you've got your mitotic spindle forming, and it's almost, what you have is duplication of your centrosome, and so you've now got 2. And they're going to attach to the microtubules and segregate chromosomes when it's time. And we'll learn about that in the next unit when we talk about cell cycle in detail. Right, so I keep talking about how we have this dynamic instability, this constant growth and shrinkage of cytoskeletal proteins, and you're probably like, okay. So I just want you to think about it a little bit more. So when we talk about cytoskeletal filaments, you have to think of it as these filaments. So that's a filament or a polymer is a group of small subunits that assemble together and grow. And so they're constructed from these small subunits. So you've got small subunits. If you look here in A, small subunits, and then a large filamentous polymer. So all that they're doing is they're assembling head to tail, head to tail. So it's kind of like building a skyscraper out of bricks, right? The bricks are the subunits, and then you assemble them into a skyscraper. The only difference is usually we don't disassemble skyscrapers all the time. We just build them and leave them, hopefully. And so what I mean, so I keep saying to you, they're dynamic and they're reorganizing in response to some kind of signal or some stimulus. So in this case, what you can see is you have your subunits and you have your polymer, right? And let's say there's some food over here where your yellow arrow is. And so the cell needs to move towards the food. And so what it's going to do is it's going to activate all these accessory proteins that I told you are coming your way, and change the situation so that it disassembles the actin polymers that were on this side, and then they become subunits, and then they migrate over here and they polymerize on the other side, and it's that actin polymerization in a certain direction that helps to move the cell in that direction. So actin polymerization on this end will help the cell to move in that direction where the food is, for example, or whatever. It could be food, it could be a bacteria, whatever it needs to move towards. Okay? And that's an important concept because I think sometimes people don't understand that all these subunits break down again and then they'll travel to the other end of the cell and grow again. Yes.
So that,
Neutrophil.
I was moving, like, just by,
So that neutrophil that I showed you that was chasing the bacteria was moving in this exact way. It basically took all its actin subunits and assembled them in one direction. Yeah, it's pretty amazing. Yeah, assembled them in one direction, so it moved in that direction, and as soon as that stimulus moved over here, it disassembled and assembled it in that direction. Yeah, yeah.
How fast does this process take the whole assembly and disassembly?
It's really fast. How fast does it go? I guess I should look up, but I mean seconds. Because if you think about it, you saw that neutrophil is chasing down that bacteria.
So that was a speed step up.
That movie was probably sped up a little bit, but it's probably advantageous for a cell to move quick if it needs to get bacteria, right?
Yeah.
But it's pretty quick. And as we learn about these accessory proteins, you'll see that all you need is to send a whole bunch of proteins that are going to disassemble it, and then boom, disassemble. So there's normally this kind of growth and disassembly that's based on the concentration of monomer available. But then there are all these proteins that will enhance that and make it faster or slower, depending on what the cell needs. I saw your hand. Yes, the accessory proteins are basically helping to regulate the growth and stability and disassembly of all of these, of actin and microtubules and intermediate filaments. Yes.
So the ones that connect to the different polymers, do they come straight to just form like a lighting section, or do they have to connect on the side?
Oh, very smart. Thank you. So the question is, just assembly, because I was giving you an incomplete story for simplicity, but you caught me. Good. I love it when my students catch me. So the question I got is this growth here enough to deal with the lagging end of the cell? Is that what you're saying? No, it's not. Because, so what really happens here is you get a certain kind of actin polymerization on one end that moves the cell forward. You get this contractile actin that forms at the other end that contracts and kind of brings up the rear. And we'll talk about that and how signaling and the Rho proteins all kind of help to make that happen. Yes?
So I don't know if this is off topic or not, but how did the bacteria induce?
How do the bacteria move? Who's taking micro?
They have the cilia,
They have cilia and they have flagella, and they actually, and then flagella are made of microtubules, right? Yeah, different, but yeah, so similar, it's like a machine made out of, cytoskeleton, bacterial cytoskeleton. So cilia, flagella, yep. Other questions? I saw another one somewhere. Now are we good? Are we happy? Isn't this fun? I love it. Okay.
So if the cell is not needed to move, does it stay in the confirmation of the middle one, or?
So if the cell does not need to move, is it all just, so this is a very simplistic way of looking at it, but we're just trying to get a concept across here. So you're saying if the cell does not need to move, is all the actin not polymerase and is all subunits? No. And what we're going to learn is there's this spontaneous assembly of subunits based on the concentration that's available. So, and we're going to go into greater detail on how this works, but basically there's a certain concentration of subunits that's going to make the subunits polymerize into a filament. And when you're lower than that concentration, you're going to get this assembly, and when you're higher than that concentration, you're going to get assembly. And so that's just happening based on the concentration. And there's a lot of actin that's always around the cortex of the cell. So it's not just that we have movement, that's just a very easy example to kind of conceptually understand. There's a little bit more than that. Any other questions? Okay, so filaments are formed from multiple protofilaments. So what does this mean? Basically, and this is really when you're starting to think about microtubules. Well, both really. And so it's not just that you're having one line of microtubules. It's not just like tubulin binds to tubulin binds to tubulin. You're actually having interactions on all four sides. So you're having lines of microtubules that bind to each other, and that provides the strength and stability and ease of assembly and disassembly. So, and you're going to see that, I think I might have, we'll see some figures that show that with the disassembly, you actually have all the different filaments kind of splay out and fall apart. So here's the intermediate filaments. I really do not spend enough time on them. I think it's because they're not dynamic the way actin and microtubules are dynamic. And intermediate filaments are mostly providing strength and allow for the stretching and bending and all that's needed when you're thinking about cells that are forming sheath, that are forming an organ, or forming your skin. You need to, if those cells just easily kind of fell apart, then we have like our skin would be breaking all the time when we poked each other. So they're forming a lot of strength. And so they're made of subunits, just like actin and tubulin. They're longer subunits, and the way they're assembled, they're staggered, and so they kind of have rope-like properties. That's what gives them their strength. And so, these intermediate filaments, they form from monomers that assemble into dimers, and then they assemble staggered into tetramers, and then form this rope-like bundle. So that's why they have so much strength. And I keep saying they're involved with formation of tissues and organs from cells. And so this is just showing one cell, and it's showing all of these blue right here, your intermediate filaments. And it's showing how it's involved with the connections between cells and the strength and the stability. Okay, so remember I said there's kind of like a language you have to learn, right? And so there's a few terms I just want you to know. I want you to think about them. I want you to understand them, because I'm going to talk about them like it's nothing, and if you don't know them, you're going to get lost. So this is the language of the cytoskeleton. So nucleation. So this is the creation of a stable starting unit that allows for rapid polymer growth. Remember, we're always and so from here on out, I'm mostly talking about actin and tubulin. And you've got all these subunits, and they kind of like will bind to each other and release, and bind to each other and release, and eventually form this stable starting unit, and then polymerize more rapidly. And I have a picture. I have a video, which is probably gonna mess things up, but we'll watch it anyways. So then there's critical concentration. And so this is kind of what I was referring to before. This is the relationship between free monomer concentration and polymer growth. So if you're at the critical concentration, then the on and off rates of monomer to your growing polymer are equal. And so let me just quickly show you the nucleation video. These videos really help with understanding this stuff. It's not hard, it's just the concept is slightly different. Oh, yeah, that's exactly what I wanted.